Thanks, everybody. Really appreciate you joining this workshop. Um, I'm Jay Famiglietti. I'm the executive director of the Global Institute for Water Security. I'm also a professor in SENS and in the Department of Geography and Planning. Welcome to the Connect the Dots internal USask workshop on research into the food-water nexus. I'll take 10 minutes this morning to give some logistics, some basic goals for the workshop, and then talk about my own research and what drives me to think about food and water to kind of kick things off. Of course, as we gather online today, we acknowledge that those of us who are in the area today are on Treaty 6 territory and the homeland of the Métis. We pay our respect to the First Nations and Métis ancestors of this place and reaffirm our relationship with one another. Okay, so just some logistics. Please join the sessions 5 minutes in advance. I know that wasn't possible today because we got a slightly late start. We'll try to do better for the rest of the day. Please try to keep your talks to 5 minutes. Stay on mute unless you're speaking or asking a question. Send your questions. I haven't looked at the chat, but the idea is to, when we do have questions at the end, so we'll go straight through the presentations. And I will moderate, and I want the other moderators to, in the other sessions, to keep track of, keeping people on time. So send your questions to Palash, or raise your hand, and Palash is going to be keeping an eye on you. I'm I will try to do the same, but it's kind of hard to do both, to have the participant grid of 50-something people, and watch the presentation. Let's, we've got an hour scheduled in between these things, and it's great to, I know we all have other work to do in the Zoom era, but it's a good time. If you see something really interesting, shoot someone an email or set up a video chat or something in the hours that we have in between. So the last session of both days, today and tomorrow, are really meant for open discussion. Big ideas, synthesis. We don't have any real particular things we want to talk about, except that we did allocate some time to talk about Bangladesh tomorrow. But in general, this is an opportunity for us to brainstorm, to think about what we saw today, to think about how we can do fun new things, and where we might complement or supplement each other's research, where we might be thinking about future hires, and it's just a way to get things going. Remember that this hopefully is the first of several workshops. We're not going to get it all done today, so the goal is really to, as I'll show in the next couple of slides, is really to get to know each other in this first session. All the sessions are recorded; they'll be posted online, and you've seen the Zoom links. They've shown up on your calendar. One link for for the different time slots. So Monday, Tuesday at 9 is one link. Monday and Tuesday at 11 is one link, etc. Okay. So hang on. So goals of the workshop are to survey the landscape of the food water research, nexus research on campus, and really, like I said, start to get to know each other. We want to catalyze new collaborations. We want to reveal and expand ongoing collaborations. I like to say that I'm still pretty new here, in the sense that I only had 18 months here before we were hit by COVID. I was just starting to get to know people. So we've been sequestered since then. So we want to build synergies on campus. We want to identify topics for deeper dive discussions and follow-on workshops. I think that's really important. I think we want to begin thinking about new projects, proposals, facilities, faculty, programs. Again, that's not all going to come from today's workshop. Today's just the beginning. And identify areas where USask is uniquely positioned to lead. So outcomes, and you'll see I get more grandiose as I go towards the bottom here. So outcomes are, of course, we'll write a report to our campus administration, to our unit leaders on our strengths and our opportunities. I just mentioned we want to have follow-up workshops. Hopefully this is the first of many that will result in joint papers and research and proposals. And then as we start thinking bigger, ideas for new faculty hires, strategic chairs, and new programs. And as I've been saying, and many of us know, we have great food research and agricultural research on this campus. We have great water research on this campus. And I think we are really well positioned to take first steps towards national, international leadership on the food-water nexus, doing some things jointly. I just want to take a quick look at everybody. I'm not looking at chat. I'm not looking at the chat box. I probably should be. Can everyone hear me okay? So I can see, okay. I see a few thumbs up. That's good enough. Okay, good enough. I'm gonna forge ahead. Okay, so I'm gonna go now to our the 5-slide dance. So who am I? I'm Jay, Famiglietti. I'm a hydrologist who develops computer models and new satellite methods for tracking how freshwater availability is changing around the world. I'm also progressively more through the last, I'd say over the last decade, become progressively more of a science communicator and government advisor. So communication to government and to the general public, especially on water insecurity and how it will drive food insecurity. And so that's been a consistent part of my message going back a few years now. What have I done? So research that I've accomplished with my team, improving the hydrology components of the land surface components of climate models. These are like the IPCC climate change models. Spent a lot of time looking at soil moisture remote sensing and understanding on the ground space-time variability in surface soil moisture to understand what it means when we look at a satellite image, what's happening within those remote sensing footprints or pixels, and pioneered hydrologic applications of the NASA GRACE satellite mission, especially in how to measure and monitor groundwater storage changes from space. And so one of the big outcomes of that work was maps like these. This is a map that many of you have seen by now, shows the world's major aquifer systems, and the trends are the colors, and the places that are red or orange or yellow are losing water. And the upshot of this is that over half the world's major aquifers are losing water past sustainability tipping points, many of them being rapidly depleted, and a lot of them underlie the world's major food producing regions. We can look here in Central Valley in California, and it's actually the lower part of the Ogallala that's really running out of water. So when you average it all up to the whole aquifer, it doesn't look bad. But if you were to zoom in on that lower part in Texas and Oklahoma and southern Kansas, it would be a bright red spot. But over here, like India, Pakistan, we were just out in Bangladesh. All of these aquifers are around Beijing. Big food producing regions. So we got water security is at risk. Since groundwater provides about 40% of the water for irrigation, water for irrigation, our food security is at risk. So what am I doing now? Trying to, about 50% administrator, 50% research. I have a research chair. So it's a mix of taking action on the last slide, and in science, policy, and communication, trying to catalyze food water research on this campus. Some examples are at a CFI for a root zone soil moisture remote sensing facility, and we have ongoing soil moisture research. I wrote an NFREF on global groundwater sustainability and have ongoing work on prairie groundwater sustainability, global groundwater sustainability, and groundwater dependent food production. And a new project exploring financial levers on the food industry, actually all industry, but food is the biggest user of water globally. Like 80% of the water that's withdrawn is withdrawn for the food industry. So exploring those financial levers to drive water sustainability. What would I like to see? Again, I get pretty grandiose here. A major interdisciplinary effort in agro-hydrological modeling. We do great hydrology modeling. I don't know what's going on the agricultural side, but I think it's time to put those two together. See, this is not everything. These are just things that are sort of motivated by my own personal interests, and I'd like everyone to do this today. A major interdisciplinary effort in digital agrohydrology, including remote sensing across scales, from the tower to the drone to the aircraft to satellites. More detailed research at the plant water interface in the root zone, new interdisciplinary water food policy research, more cross-campus initiatives like GIFT's Bangladesh initiative, more USask-led reports and studies of important food water topics like our GIWS preliminary assessment of the Saskatchewan irrigation project. Very strong USask scientific leadership on these topics, including science communication and policy advising, and might as well set the bar high, a world-leading food water program at USask. Okay, here's our agenda, and I am now, we go vertically here. So looks like Lingling Jin is up next, and we'll go from there. And I will try now to pass the virtual baton pass, or Palash, can you handle that, or do I have to do that? And just excuse our, logistics here.
I can take care of it. Sure.
Okay, so Lingling, you're up.
Thanks, Jay, for the opening and introduction. Can I share my screen? Host disabled participant screen sharing.
Lingling, you can now share.
Okay, it works now. Can you see my screen now?
Yes.
Yeah, okay. Hello, everyone. I'll start from my introduction. Okay. My name is Lingling Jin. I'm an assistant professor of computer science. My background is in computer science, and I'm trained in the field of bioinformatics, which applies computer science foundations and technologies to biological data modeling, prediction, and analysis. After finished my degrees, I worked at AEFC Saskatoon with Dr. Isabel Perkin. Then I was an assistant professor at Thompson Rivers University in BC for 2 years, where I had the opportunity to get involved in some environment science and data science projects before moving back to Saskatoon. My research concerns computational modeling of various biological systems, as well as their interactions and how they evolve using statistical models and machine learning approaches. I'm very excited to work with people from various aspects of plant science, plant biologists, physiologists, breeders, plant pathologists, and also molecular biologists and microbe ecologists. I found a great pleasure, and it's also a very rewarding experience to work in this area. And thanks, Jay and Palash and folks behind the scene for organizing the workshop. What I have done in the past, we have developed algorithms to predict ancestral flowering plant chromosomes and gene orders, and applied them to genomes from a broad range of monocot orders, confirming the time of occurrence of the whole genome duplication event, tau. And we also modeled genome rearrangements within the Brassica group. We have successfully reconstructed the ancestor and also unraveled the rearrangements distance between the 3 diploids of the use triangle. And also developed models to study the interactive activities of transposable elements throughout time by extracting the patterns of their remnants in a genome. And this is the interaction network of transposable elements. On a practical side, the software pipelines we developed are publicly available for pangenome construction, which captures the entire gene repertoire of a species. And we also developed a comprehensive GWAS pipeline with cross-validation by Lasso, and a structural variance detection with cross comparisons and several ancestral genome reconstruction packages using various methods. The tools and the methods that I used in this work, including but not limited to machine learning approaches such as hidden Markov models, stochastic context-free grammars to model the probabilities of interactions, and clustering methods to form ancestral chromosomes based on the adjacencies. And various graph algorithms because we abstract biological problems as graphs, and statistical models to find and assess associations between and the prediction performance of the results. We also used a Bayesian hierarchical model to assess the effects of COVID-19 on students' performance in the past term. And also adjacency and co-occurrence analysis, computational optimization, and linear ordering problem. And there are several ongoing projects that I'm involved in, including reconstruction of ancestral subgenomes before and after whole genome triplication in flowering plants, and another projects on metagenome and metabolome-wide association studies to evaluate the effects of crop rotation and see how the associations with the wheat fusarium head blight resistance in terms of microbial and metabolome perspective, and also discovered the knowledge embedded in gene set enrichment analysis. And like this is environment science to study the interactions between soil, microbe, and plant to enhance mine tailings reclamation in northern BC. Don't have time. What I like to see in food and water. And these are a few preliminary ideas on the food-water nexus from bioinformatics perspective. And I think we could ask questions on like how to resolve plant species, how plant species have evolved and identify their shared evolutionary pathways related to adaptive traits, and also study the associations between root zone soil moisture and soil microbe evolution related to soil water profile, plant responses, and the genomic features of the plants. We could also use computational genomic tools to aid in rapidly selecting genetically drought-resistant crop ecotypes, and tie multi-year crop growth stability and characteristics to genomic features and hydrological features of the farmland. I'm still very new to the topic of food and water nexus, and I'm keen to learn more problems from the workshop that I could contribute to. Thank you very much.
Okay, thanks, Lingling. And so for those of you who are not on mute, please go on mute. There's some background noise. And next up is Bobbi.
Sure. Okay, can you see my slides?
Yes.
Okay, terrific, thanks. Sorry, I'm having a little confusion. Let me just, sorry about this. Oh, gosh, sorry.
No problem, Bobbi. You know, are you? Would you like us to just go to the next speaker while you sort it out? I mean, or are you like, it's no problem. Okay,
There we go.
hang in there.
Terrific. Sorry about that, everyone. All right, so good morning. I'm really happy to be taking part in the workshop. My name is Bobbi Helgason, and I'm an associate professor in the Department of Soil Science. So I am a farm kid from Saskatchewan. In the background here is a picture of the farm where I grew up. For the last, from 2010 to 2018, I was working as a research scientist at Agriculture and Agri-Food Canada, and I more recently joined the University of Saskatchewan in the fall of 2018.
Bobbi, sorry to stop you there. We can't see the slides.
You know what, why don't you go to a different presenter, because I'm looking at them and you're not. So if we can do that, we'll try to sort this out on this end.
Yeah.
Okay, no
sounds
problem.
Sorry, everyone.
No worries. So Haben, are you ready to go?
Yes, I am.
Okay, let's go for it.
Okay. Let me know if you can see my screen.
Yes, we can.
Sure. Okay. So my name is Haben. I am a research scientist, recently joined AFC as a systems agroecologist. Yeah. So I have 5 years of experience in precision agriculture before I joined AFC with a private industry known as Farmers Edge based in Winnipeg, actively working across the prairies and US and other places. Before that, I was a research associate at the University of Manitoba as in a soil science department, as well as in animal science department, mainly working with manure and fertilizer, nitrogen fertilizers, and looking at greenhouse gas emission. And my background is in soil science, mainly in soil fertility and plant nutrition. Graduated from the University of Bonn in Germany. And I was also working in ICARDA, one of the CGIAR centers. At that time was based in Syria. And my master's degree is in agricultural sciences and resource management, mainly with plant nutrition, and bachelor science in soil and water conservation. And I like soccer, walking, and reading. So I can define myself that way. Tools I'm using at AFC is I'm developing the lab from scratch. So I am purchasing softwares like the SMS Ag Leader, and also I do have the ArcGIS Pro. The SMS Ag Leader, it's for variable rate application. You map out zones, and you can manage also different data, spatial data, using that software. And also I just acquired the Synergy H1 micro reader that will help us to determine enzymes like phytase. So just delivered, and we are also on purchasing Picaro, but we don't have the GC for a long time. And Rainfall Simulator and HyProp are the new equipments. I do use lots of R packages like HMR, statistics, and dominance analysis. Many packages in there. So what I have done so far is I have a lot of experience with variable rate application of nutrients and irrigation. That was from the industrial experience that I had. And also I was working for a long time on nitrous oxide emission and developing new coefficients for nitrogen fertilizers and management systems, like placement, timing, stuff like that. I had also a lot of work on nutrients and water dynamics across transects, and also quantified nutrient balances at different scales at farm, field, and community level of quantification methods. Now I'm working more on mitigation of nutrient losses in collaboration with Jeff Shannon on his project, actually, on precision manure applied fields. And also I'm working on developing quantification methods of resilience, crops resilience to biotic and abiotic stresses. We do have a big project in 7 sites, and in looking at comparing 6 cropping systems, and how they are resilient to biotic and abiotic stresses. New projects are on soil nitrogen residual, especially how to look at them. And I am hoping that to get funding for applied in phosphorus and nitrogen and how to mitigate them. And I'm hoping to use the mine overlay facility. I would like to see a multidisciplinary research which targets shorter and long-term scenarios, and also looking at the crop productivity and water use efficiency while maintaining higher environmental standards. So keeping the nutrients in the field and reducing losses. One big question for the group is to consider to add energy on the food and water nexus, and also to consider environmental issues like water quality and mitigation of greenhouse gas emissions. Yeah, thank you. That's what I have.
Okay, thanks very much, Haben. Bobbi, are you ready for us to revisit you?
I think so.
Okay, let's give it a try.
I just got host disabled screen sharing for a moment here. So.
You got it.
Is there someone that has to let me in?
Yeah, you are the host right now, Haben.
Yeah, it's Bobbi Palash.
I'm blocked out here.
Scott, we're trying to send you a message, Bobbi.
Okay. Oh, sorry. I need to make Bobbi. Yes. Give me one sec, Bob.
I promise it's going to happen really soon.
And hopefully I can get it right this time.
Okay.
There you go.
All right.
All right. There we go. Are we on? Are you able to see my screen now?
Yep.
Terrific. Okay, thanks for your patience, everyone. So good morning again. I'm Bobbi Helgason. I'm an associate professor in the Department of Soil Science. I got my PhD in 2010, at which time I joined Agriculture and Agri-Food Canada as a research scientist, and more recently the University of Saskatchewan in the fall of 2018. I was brought on through the GIFTS operated Plant Phenotyping and Imaging Research Center, where together with Steve Siciliano, I co-lead the soil root microbe interactions flagship. In the background here is a picture of the farm where I grew up. It's near a little hamlet called St. Gregor, Saskatchewan, about 150 kilometers straight east of here. And that pond behind the yard is actually part of the Quill Lakes waterway. And so I put this up just to make the point that actually as a kid, my parents were small grains and oilseeds farmers, and water in the landscape was a big part of what of their farming operation. Oh, okay. Okay, here, let's do this. So now we got a picture of the farm where I grew up. And so all of Mom and Dad's land is actually engineered to be drained by overland V ditching. And so too much and too little water on our farm was certainly a big part of the story growing up. I'm a soil microbiologist. This is my team. And I like to think that I really try to, through collaborations, provide some of the how and why when we're unpacking below ground processes of soil nutrient transformations and plant productivity. So the major themes of my research area are soil carbon stabilization and destabilization. Microbes are agents of decay. They take plant residues, decompose them, and stabilize them as soil carbon, driving CO2 losses from the landscape in the process. Nutrient cycling and prevention of losses with a focus on nitrogen and really nitrous oxide emissions. I've done some work. We've got students currently working on nitrous oxide emissions and carbon cycling in pasture systems, as well as from canola production systems. And I also do plant microbe interactions. And so this work is largely driven through the Plant Phenotyping and Imaging Research Center. In terms of the tools that I use, opening the microbial black box, there's enormous below ground biodiversity, but capturing and describing that biodiversity is definitely a challenge. We use microbial community profiling, accessing the identity and the abundance of microbes below ground through their nucleic acids, their DNA, their RNA. We also use a tool called phospholipid fatty acid profiling. But ultimately knowing who's there is just part of the question. And so we spend a lot of time looking at soil functional assays. So the activity of microbes below ground through things like soil respiration, enzyme activity, nutrient transformations from one form to another. And one of the special tools that we use a lot in my program and at the University of Saskatchewan is stable isotope tracing. So the use of carbon-13 and nitrogen-15 as tracers so that we can follow carbon and nitrogen, for example, from the atmosphere as CO2 through the plant in the process of photosynthesis and below ground into different pools. So in terms of our discussion today, I think I see special links in the food-water nexus realized through a better understanding of the role of soil organic matter in agroecosystem services. So more recently, my research program has been focusing on the right hand of this slide, or pardon me, the left hand of this slide, looking at the role of roots in soil carbon contributions, turnover, and stabilization. But ultimately, soil carbon questions are very simple. Input minus output equals storage. And so plants capture CO2 from the atmosphere, they fix them in their tissues, put them below ground, and microbes as agents of decay transform them ultimately into stable soil organic matter. And we know that water is one of the most limiting factors for plant growth on the prairies. And so it certainly plays a special role in understanding how to improve soil carbon stocks and storage. And so I think that this is a picture of a landscape taken last June near Bigger. And so what we can see here is varying levels of soil productivity across the landscape, really driven by water dynamics. So dry, unproductive knolls are lighter in color, and depressional areas where water is less limiting are darker. And so how can we capitalize on the infrastructure and the talent and the research capacity of both the Global Institute for Food Security and Global Water Futures, perhaps through things like remote sensing and deep learning, to bring together these processes occurring at broad and very narrow scales of space and time. So what's the microbe perspective, and how can we link that to things like remote sensing across the landscape to better understand landscape scale processes? And so I think I will wrap it up there.
All right, glad we could get that worked out, Bobbi. So Peter, you're up next.
Okay, I need host, please. There
Peter,
we go.
you're the
Thank you.
host.
Yep, thank you. Got it. And I assume you should be able to see my screen now. Can you?
I can't hear you very well. Palash, how well can you hear Peter?
Can you hear me now?
A little bit better.
This way, I sometimes get feedback if I don't use the earphones.
Okay,
Is that okay?
that's much better.
That better? Okay, can you see the screen? I have
Not
shared
yet.
it. I've
No,
shared
don't
it.
see. We're only seeing. Oh, it's coming now.
Okay, good. Okay, there we are. My name is Peter Phillips. I'm a professor in the School of Public Policy, and I straddle the science policy interface. I spent about a third of my career in the public service, so at senior levels of policy advising. So I've dealt with science as policymakers and as politicians explore and try and figure out how to use the work that you produce. My approach is fairly eclectic. I'm trained as an economist, but with political science overlay. And I use a mixed set of tools that draw from across the social science spectrum. Institutional analysis, which is pretty standard, looking at who has power, who has authority, but added some of the qualitative tools, looking at texts and how people act and record the results. But we do some modeling, we do some surveying. I have a piece of the experimental decision lab where we actually test your cognitive capacity to think in well-ordered ways that leads to predictable outcomes. We do some social network analysis.
I don't want to take that test.
We all fail, even the ones that know the answers. We do some social network analysis to understand systems like you're trying to build today, about how they function and where the strengths and weaknesses are. And then we sometimes take that into the in silico world and do agent-based modeling of research space to understand how design influences output and impact. So what have I done? This bundle of tools is largely focused on the bioscience innovation space, with a particular interest in how that contributes to global food security. So my team has explored an array of issues around that, how governments organize science, tech, and innovation policy. And that involves things like clusters, innovation systems. It involves the governance of the research spaces that we live in, how the tri-agencies and others try and incentivize us to do things. The role of big science, labs like the CLS and the Vito Intervac. And of course, as we move downstream, you produce things that have potential value, some of which are protected through formal property rights, some which bump up against the Indigenous rights embedded in access and benefit sharing and traditional knowledge policies. We do look at how some of these new technologies, or more appropriately, the products of the new technologies, are embedded in regulation, and how their products are introduced into the marketplace, both for consumers and for the industrial supply chains. So this is much more of a food focus than a water focused. And ultimately, these things are traded. Everything that comes out of Saskatchewan goes somewhere else in the world, either interprovincially, internationally. And so trade and market policy and strategy are core to how the system works. So my basic question, and it's in PERC and it's virtually every other product, is that scientists have wonderful visions of how they're going to change the world, but they have a differentially, the ones I worked with, have a field of dreams mentality, that if I build it, they will come. All I need to do is build the diamond and get the lights up, and the teams will move in and occupy and use the space as intended. And if there's one thing about innovation studies, it tells us that almost never does a product of a lab or a scientist or even an entrepreneur end up being used exactly the way it was intended to be. It was designed to be used. We turn it over, we turn it upside down, we apply it in different spaces. And so the question is, under what conditions does it have a chance of getting used? And then secondly, under what conditions will it get repurposed and realigned, and so that it adds the optimal value to the economy and society? So in my space, the big question is, how do you integrate the policy design across the two domains to explore both the trade-offs and feedback loops that exist in this area? Water with wetland conservation and resource management, interfaces with plants, with new water efficiency varieties and crops. There's precision ag, which is another tool that farmers individually are using to manage their risks through rotations and precision inputs and management of outputs. And then there's all the policy levers. Could be crop insurance, land use, water pricing, water allocation rules. You may, as a scientist, say, I've got the better mousetrap, but you're competing against at least three other domains where there are credible alternative options. So the challenge is, how do you build that into how you think about where you allocate your resources, how you motivate your research for preparation for use. And so the big question, I think, for the group is, how do you know that when you promote, propose, fund, staff a research effort, that the insights and the innovations you seek will actually influence something? They'll influence the choices we make, either structurally or in procedural terms, for the allocation and use of water, soils, and plants for the future of Saskatchewan, and ultimately for the future of the world. So this is really the big impact question. And that's why I spend a lot of my life embedded in science teams, trying to explore what they're doing and figure out how can we improve the potential for impact. Thank you.
Okay, thanks very much, Peter. Melissa.
All right, just a second. Okay, you can see me, everything's good? All
Yeah,
right,
looks good.
perfect, thanks. So I'm Melissa Arcand. I'm an assistant professor in the Department of Soil Science here at the U of S, and I've been here for about 5 years. And prior to that, I was a postdoc at Ag Canada with Bobbi Helgason, and I did my PhD here in the Department of Soil Science. So who am I? I'm a soil biogeochemist, and my research really focuses on looking at the biological mechanisms that control nutrient and carbon cycling, and broadly at 2 scales, essentially. So more broadly, looking at the landscape scale and across agricultural systems. So I've done work comparing organic versus conventional systems on nutrient cycling and carbon storage, as well as in pasture and grazing systems, and looking at cover crop systems. So broadly looking at those controls on nutrient carbon cycling that will ultimately affect soil health. I then do work that zooms in on a much finer scale, looking at root-soil interactions, again on nutrient and carbon cycling, but with the intent of looking at how roots might input carbon to the soil and how that carbon might be stored, as well as looking at crop nutrient uptake and nutrient use efficiency. Along with that, something about me, who I am, is I'm also an Indigenous scholar, and I've got both research and teaching collaborations with First Nations communities here in Saskatchewan. So what are some of the tools and methods that I'm currently using or that I've used? So using sort of classic methods in soil biochemistry. So often looking at extracellular enzyme activities that control nitrogen and phosphorus and carbon cycling, and as well as some of the tools that Bobbi talked about in microbiology. So who's doing who's controlling those processes? Something that's foundational in the work that I do is the use of stable isotopes. So both carbon and nitrogen stable isotopes to look at how both plant roots and crop residues might be processed within the soil, and what their fates might be in the soil, and also potential losses of those nutrients to the atmosphere. From the root and rhizosphere research that I do, I'm also employing root imaging techniques, both in situ techniques such as mini rhizotrons, as well as more classical in controlled environment studies, doing flatbed scanning. And then some of the work that I'm doing in collaboration with Angela Arcand is also employing digital soil mapping and GIS to the more broader landscape scale research that is more new to my research program. So what have I done? Again, focusing on stable isotopes, I've quantified nitrogen and carbon inputs from crop roots into soil. And this has provided insight on, for example, nitrogen budgeting in pulse crop systems, so we can get a better handle on the contribution of nitrogen from legumes that can use atmospheric sources of nitrogen, and where that nitrogen ultimately goes in the soil. I've looked at biological indicators of soil health in agricultural landscapes and across different agricultural systems, as well as looking at nitrogen cycling in the rhizosphere and crop nitrogen use efficiency with a focus on canola. And also in shifting towards more social science realms, also as a PI on a forum on Indigenous agriculture in Saskatchewan, and I recently published that work. And so I'm going to talk more about, in terms of what I am doing, moving further away from my classical training as a soil biogeochemist, and looking at ways in which I can apply that research, specifically in working with First Nations communities. And so currently I've got a New Frontiers and Research Fund project working with 2 First Nations communities in Saskatchewan, where we're conducting soil health research on agricultural lands, and especially examining the effects of long-term leasing on their soils. And this has been an incredibly collaborative project, engaging the lands departments for both First Nations communities in identifying specific research questions, as well as in collaboration with Indigenous studies scholars who have the background and methodology in doing some of this community-based research. So specifically,
Hello.
hi. Sorry.
I would just keep going. It sounded
Okay.
like someone just saying hello.
I guess so. That's a friendly interjection. So hello. So what would I like to do and see in this food water nexus, I suppose, is I'd really like to be able to extend the work that I'm doing in soil health in First Nations agricultural lands to collaborate with experts in water quality and wetland health. And ultimately also be able to engage researchers who are in the human health realm as well, especially in this age of pandemics and trying to mitigate risks around that. How can our work better address especially food security and water security? So I guess that's my big question for the group, is how can natural scientists better engage with Indigenous community-based research to address issues of food security and water security? So with that, I guess I'll just end there and look forward to the discussion that we'll have later today.
Okay, thanks very much, Melissa. Clint.
Yes, hello. I'll try to share my screen here. First time doing so on Zoom. Uh-oh.
Oh, so while Clint is trying to figure out how to share the screen, we can see that his hobbies are reading and playing guitar.
Yeah, that's right. Is there any way somebody can walk me through this? I've never done this on Zoom. Okay, Microsoft PowerPoint, that would be it. You can't see this, can you? Or just my security and privacy thing?
We can only see you. We're seeing your face, your books,
Okay.
and your guitar.
All I have is photos, so I'll talk you through it, okay? I'm a social science researcher. I'm an environmental anthropologist. I've been working at University of Saskatchewan since 2009. My graduate training is in anthropology and environmental studies. In terms of tools and methods I use, I'm primarily an ethnographic researcher. So I've looked at different aspects of First Nations culture and history in Northern Alberta. Basically, I'm applying qualitative methods and I'm directing a research team called Cultural Policy of Energy, Cultural Politics of Energy in Northern Alberta that has had SSHRC funding since 2011 or so. And so currently we're working with a variety of partners in community engaged research. And the team includes experts from anthropology, sociology, earth sciences, and Indigenous studies. And so one of the rubrics or titles we've hit on is Eating in Oil Sands, which is the title of a PhD thesis from a colleague in the research group. And it seems to bring together things nicely in the sense that people are still using the heavily industrialized environment in Northern Alberta to extract a lot of their food needs and of course water. Recently, research has shown that wild foods are becoming a vector for contamination into people's bodies. Many people in these northern communities are eating a significant amount of wild meat. And the way that the contamination gets into the animal bodies is largely through water. So right there, you see a really nice illustration of the food water nexus. Some of the things I've been doing, trying to transition more into working with scientists, but also critiquing the science that's being done in the form of impact assessment statements. Impact assessment is an industry that must incorporate social considerations and particularly Indigenous rights to harvest wild food. So this is sort of one of the points of contact between the regulatory mechanism and consultation in communities. And so we've done a lot of critiquing of the types of assumptions that are put into this and really critiquing it from a social science vantage point. It's only recently the social science of oil sands impacts is catching up with some of the natural science, and the natural science also kind of lagged behind until the industry had expanded a great deal and the horse had kind of left the barn. So now we're working with First Nations to determine impacts from various types of projects, both intensive and extensive, depending on different technologies to access energy. Also looking at the connection with forestry. So there's cumulative impacts from a large number of industries. And I've been working in Northern Alberta for about over 20 years. I would like to see from at U of S or more broadly in water and food research, I guess I'll stick to U of S, is to bring in more and develop more of a qualitative lens on social science that looks at lived experience and tries to evaluate things in a way that goes beyond social hydrology or counting volumes of water, and looking at how people engage with food and water in a very intimate way that touches on their health, culture, food and water sovereignty. And so it's nice to follow up on Melissa's work and Peter Phillips. Peter, whom I've only met virtually, and Melissa not having met at all. And so trying to think about representing the full range of social science research, touching on food and water here on campus. I don't think we're quite there yet. And also the question I have for the group, I guess, is to think about how can we bring ideas of lived experience, culture, religion, spirituality, ceremony, and things of this nature into the way we study food and water, so that it connects with people's lives and personal understandings on a very real way. So I'm sorry that I didn't get my slides up. I didn't follow the template anyway. So hopefully this will help you understand a little bit of the kind of work that environmental anthropologists do. I have a lot of photos of people hunting moose and also some ugly pictures of scarred post-oil landscapes. So I'd be glad to share those with anyone who's interested. I'll sign off now, Jay.
Okay, thanks very much, Clint. Maybe it was better, although the pictures sound great. I found it a little easier to focus on what you had to say. So who knows, maybe we'll just ditch the PowerPoint slides. That said, if you could send them to me, if all the, I'll send out an email after the session, but I'd like to collect all the PowerPoints to refresh my memory. You know, my note taking is only so good. Thanks very much, Clint. Jian Liu is next.
Yes. Hello, good morning, everyone. So my name is Jian Liu. I'm a postdoctoral research associate at JWS. And I actually work with Helen Borch at JWS and Jian Liu at Environment and Climate Change Canada, and also partly with David Laub at the University of Manitoba Soil Science Department. So I got my PhD from the Swedish University of Agricultural Sciences in 2013, and after that I did my postdocs in China at the Chinese Academy of Agricultural Sciences and at the Penn State University in the United States before coming to Canada in the December 2017. So I'm a soil scientist. My research is focused on understanding nutrient cycling in agroecosystems and assessing agricultural management and climate impacts on water quality and also crop productivity. I think nutrients are really important because they are important for crop production, but if they are lost from the agricultural production systems to the surface waters, they cause eutrophication there, and they have a lot of negative impacts on the ecosystems in water. I work on different scales. So in the lab, I conduct quantitative measurements to determine how soil properties, plant properties affect their ability to affect like for example the phosphorus absorption, desorption processes, and how that could affect nutrient losses from this fine scale. And in the field and watershed scale, I work on this kind of agile field monitoring technique, and to monitor water quality and to link the water quality to climate, soil, crops, and nutrients, to look at how those factors or drivers affect nutrient runoff or leaching. And I also work with meta-analysis based on published papers, etc. In the past, I also worked with nutrient leaching experiments, both in the field and in the lab, and also runoff experiments with simulated rain in the field. And I also worked with watershed modeling with the tool is called Soil and Water Assessment Tool. It's a model developed by the USDA ARIs. In the past, I have assessed how nutrient source, application rate, timing, and placement affect soil fertility, crop yield, and water quality. And I build relationships between phosphorus application history and soil test phosphorus and phosphorus runoff and leaching. And also I assessed agronomic and environmental trade-offs of cover crops in cold climates. Because on one hand, cover crops could remove nutrients from the soils and they protect the soils from erosion. But on the other hand, in cold climates, they can become a source of nutrient loss, because after freeze-thaw process, the nutrients will be released from the plant materials and they can end up in runoff. And also I worked on improving field and watershed models and apply them for assessing management and climate impacts on water quality and crop production. And also I have led some collaborative efforts. For example, I led to publish a special issue on agricultural water quality in cold environments last year. Currently, I'm working on synthesizing the water quality data that are collected from Saskatchewan, Manitoba, and Ontario at multiple sites and years. Actually, we have over 200 sites and years of data, and I'm analyzing the data to look at how climate and management impacts water quality. And also I'm working with David Laub on a project to look at how crop impacts, how different types of crops impact rain interception, snow trapping, and as a result, how they affect runoff generation and nutrient losses at the field scale. And also I work on to look at how agricultural drainage and ditch processes, like for example, vegetation, how they impact water quality in Saskatchewan. And I'm also organizing a workshop on agricultural and environmental phosphorus management to try to explore the best management practices to benefit both crop production and water quality. And this is a cross-province effort. So the organizing committee included the people from Saskatchewan, Manitoba, and also Alberta. So I would, from this workshop or the continuing effort, I would like to collaborate with agronomists, crop scientists, and hydrologists to better understand nutrient and water cycling in the plant, soil, water, air system, and explore interdisciplinary measures that can be used to improve water quality without impacting crop yield. So basically, I'm interested to explore the measures that could be used to lock nutrients and water in the soil. And I'm also curious about if from the, for example, JIFS, if there are research about low phosphorus demanding crops or high phosphorus removing crops, if these crops can be applied to the fields so that we can reduce the phosphorus application rates or remove phosphorus from the hot spots. And also I'm interested to look at if there are any frost resistant cover crops, because the cover crops are increasingly used and they can become a source of nutrient loss. But if there are species that can resist frost, then they would be a very good species for Canadian agriculture. So my big question is, can we improve Canadian water quality while increasing or maintaining crop production? Thank you very much.
Okay, thank you very much, Jian. So we are past our 10 o'clock. What I'd like to do is still have 10 to 15 minutes of discussion. If you can't make it, no worries. But that is in part why we left time between the sessions. Also, feel free to contact each other. For the next session, please do your best to stay within 5 minutes. I know it's difficult, but consider it a challenge. And don't feel like you have to tell us everything that you've done. And you know, one strategy might be to look at your slides and cut your bullet points in half. Again, this is the first of many meetings. So one thing I'll say right from the start, I thought that for me personally, so there were a bunch of people that I didn't even know existed, and I wasn't likely to find because of COVID. You know, not likely to without something like this workshop, I don't think I would have been likely to bump into you for a long time. And so just in this one subgroup, I learned a lot about people and interactions. For example, you know, there was a lot of strength there that was evident in the soils, whether we're talking about microbes, roots, nitrogen, water. So I thought that was really great. There's a lot of strength there. A lot of course, you know, on our campus, a lot of interest in research and engaged research going on with Indigenous communities. But I really like some of the comments from Cliff and Melissa and Peter. There was a recurring theme that I think we all want to figure out how to do this. How can we grow more food? You know, how could we do it with using the least amount of water, the least amount of nutrients in ways that are environmentally friendly and culturally respectful, and that engages the food industry, or we were even talking about the oil sands, you know, engaging industry in environmental stewardship. So that's a big one. That's a big one as well. I think if you have any questions, I want to see who's still with us. So why don't we, if you have some questions, shoot them into the chat, and we'll see what comes up. And if nothing comes up, I may ask a couple of questions. Okay, I'm seeing no questions pop up. I'm going to take a look at the gallery.
Leon has a question, Jay. Leon has raised hand.
Okay, I don't see that. Oh yeah, there's Leon. Nice. Leon, what's your question?
It's not a question, just a comment kind of following on yours. you know, like Jay and I are, you know, newer to the campus than most of you. I've been here about 4 years and Jay too. But I thought after 4 years, I know most of the people here. This first group, there's about half of them I didn't know, particularly more junior people. And I really, it's interesting. I mean, I was worried that so many speakers I was wondering how this would come off, but I thought this was pretty successful first session, maybe a little long, but just for example, Jian Liu, I didn't know you had somebody working in that area, which actually overlaps, just getting away from the water part, the phosphorus part. You know, we're developing crops that are more efficient, use less phosphorus, and that's really important for developing world. But just another example of potential for interactions that could grow out of this that may be directly at bearing on the food water nexus or tangentially. So just a follow on comment to yours, Jay.
Thanks, Leon. Anyone else? Jeff wants to say something. Hi, Jeff.
Hey, how are you? Yeah, just
Great.
a quick comment that I too really enjoyed this session. And, you know, when I think about intersection of things I heard with Bobbi and John and Melissa and Haben, and how it resonates with things that I'm doing and others will hear from today, you know, mechanistic assessment of the water balance at kind of the pedon level, space-time aspects of root water balance and how that links to nutrients. These are really rich areas that I think, you know, as the day goes on and through tomorrow, I think, you know, we might find some exciting new areas of collaborations. And I know that's yours and Leon's goal with all this. So I just wanted to, you know, make that very brief comment.
Thank you. Thanks, Jeff. You know, I feel like we're going to be very quickly overwhelmed, right? I mean, because just this one session was pretty rich. So with that in mind, of course, we're going to make all the PowerPoints available for all the speakers. I'll send out an email afterwards, but if you're on still on Zoom, please send me and Palash and Hassan your PowerPoints. It might actually be easier for me to go through your PowerPoint than to go through the recordings, but man, that was pretty rich. Okay, so it looks like we're busy patting each other on the back. One question to well, I can see Jeff and Leon, so I'll ask you guys. Do you think that we should? So I don't, you know, I want people to go, you know, try to limit their presentations to 5 minutes, but basically I think we can expect that everyone's going to fill up the hour. So do we want to just say, okay, we're going to take an extra 15 minutes discussion, just acknowledge that? What do we think? Or do you think people want to just get off at you know, at the hour?
Well, I think we can continue to encourage people. Like, I haven't put my presentation together. I'll make sure to, I think it's a good point. You have your text slides, cut the bullets in half. I mean, too much text is always a killer.
Yeah.
But, you know, I guess from my point of view, I'd be happy to, I think it's important to have the discussion, and I'd be happy to make these hour and 15-minute sessions.
Okay,
you know,
so why don't we,
if needed.
Yeah, and again, I think we still have time for discussion at the end of the day. We're not trying to do it all. I mean, we're trying to get to know each other. One reason for taking the breaks is so we can refresh our brains and not have to stare at the screen, which I think by you know, by 4 o'clock we could all be toast. Anyone else have any comments or thoughts or questions?
I agree with Leon. Extending it to an hour and 15 minutes sounds like a great idea. Having opportunity for discussion would be great.
Okay. I'm just looking for people waving at me or raising their hand in the chat box. Okay. Well, I think this is probably a good time to break, since there's no, we've had a successful first session. So I can't see if Andrew is on here still, Andrew Ryerson, or if he's not coming on until the next session. So I'll just chat with him before we start the next session. Okay, so let's take a break. relax your brains, relax your eyes, and we'll see you at 11 o'clock.